Today I'm going to talk to you about my PhD project entitled Unraveling the Molecular Mechanisms of Monoaminergic Neurons Development through an RNAi Screen. First of all, I want to introduce you the main character of this project, which is C. elegans. This nematode is hermaphrodite and its reproduction is produced by laying eggs. And as you can see in this short video, this is a C. elegans embryo that its cells are dividing through the 9 hours that last this period. And as you can see, we can be able to track every division that leads from the progenitors to the cells that will form part of the adult worm. So this means amazing, right? And that's why we use this animal model to understand how, from this embryo, the cells acquire their terminal fate. But we cannot study all the cell types in the worm, so we focus on how do neurons in this animal become neurons. And in my case, I'm interested in the monoaminergic neurons, which are in charge of a plethora of behaviors such as movement, learning, appetite, and emotions. But the disbalance in this neuronal subpopulation can lead also to several diseases such as Parkinson or schizophrenia. And in this worm, we found 4 pairs of dopaminergic neurons. We found 5 pairs of serotonergic neurons, and also 1 pair of dopaminergic, another pair of octopaminergic neurons, which are like the orthologs in invertebrates of noradrenergic and adrenergic neurons. So the maturing of these neurons is characterized by this battery of enzymes and transporters that what we call the monoaminergic pathway genes. And we are interested in how a neuron expresses all those genes. And if you pay attention, all of them share the expression of the monoamine transporter. So, and as I mentioned, for a mature neuron, it needs to express a battery of enzymes, transporters, and this expression is regulated by a battery of transcription factor that binds to the promoters of all those genes and regulates their expression. So what we want to do is to optimize the RNAi strategy to perform an RNAi screen against all the transcription factors of C. elegans. And finally, with this process, we can also affect the lineage of the cells. And we already know that we affect the lineage because it's very good described in this animal model. So we need to validate who of those candidates are playing their role in the terminal features of the neurons. So what we finally want to do with this neuronal subpopulation is identify the specific combination of transcription factors that lead to the development of each monoaminergic neuron. And as you can see in the picture, if we call those combinations of transcription factor like terminal selectors by the role they are playing, we finally want to determine the terminal selector code of each monoaminergic neuron. So as was explained in a talk before, to do RNAi with C. elegans is very easily doing by feeding, because you feed the worms with the bacteria that express the double-stranded RNAi. We have a library against all the transcription factors and C. elegans, and the time course of our experiments would be that the first generation that hatch and start feeding on these bacteria would be our P0 generation, that when we do the experiments give us information about the importance of this given transcription factor in the maintenance of the phenotype of these neurons. But if we choose to do the experiments and the scoring with the second generation, the F1, we obtain information about the development. So we need also a strain to do the experiments, and we work in a mutant background because the neurons need to be sensitized to these RNAi effects, and also we need a reporter to score the neurons. We choose the CAT1 promoter that is expressed in all the monoaminergic neurons, fused to GFP, but also another marker that labels the dopaminergic population and another one that labels the serotonergic population, the 3 reporters in the same strain. So, as an overview of the results, there are like 876 transcription factors and C. elegans that can be organized in these 54 transcription factor families, and we do the scoring against all of them. We did 2 technical replicates, and finally we considered the candidates that present more than a 10% of phenotype in both replicates. That gave us 76 candidates. And now we need to validate the results, but we need to focus in the best candidates for each neuronal subtype. This would be the best 5 for the dopaminergic subtypes, this one for the serotonergic population, and those ones for the triaminergic and dopaminergic neurons. So we need to, we decided to start the validation experiments with a candidate for the ADF neuron, which is a chemosensory neuron that has a sensory cilia which is in contact with the environment. And when we did the screen, we observed that a member of the HLH transcription factor family, we call HLH14, gave us phenotype when we scored the P0 generation and also the F1 generation, which means that this transcription factor is involved in the maintenance and also in the development of this ADF neuron. So, as I mentioned, ADF is a chemosensory neuron, and as it is in contact with the media, we decided to do calcium imaging experiment to test the ability of this neuron to send changes in the media without this transcription factor. So we decided to give a stimulation protocol of 4 pulses. As you can see here, these are the response of the neuron when we give the 30 milliseconds stimulation with a buffer that has copper and pH 4, which are the chemicals that respond this neuron. And this is the control conditions, but when we test the worms after feeding on HLH14 RNAi, the response is completely lost, and as you can see in this red line. So HLH14 is required for ADF to accurately respond to changes in the environment. And as conclusions with this screen, we have adapted the RNAi strategy to get information about transcription factors involved in the development of monoaminergic neurons. We get information about genes that are involved in their cell lineage, in the migration of these neurons, but also the ones that we are most interested in and in terminal selectors. For the ADF, we saw that this transcription factor is not only important for the serotonin synthesis, but also for its sensorial function. And finally, we hope that with all these results, we can shed some light in the diseases related to monoaminergic neurons. And thank you, all of you, for your attention and for the funding, and especially for my lab mates.
¿Preguntas?
Gracias por tu charla.
Gracias, no te veía.
Gracias por tu charla, que ha sido súper interesante. A lo mejor lo has dicho, yo no lo he pillado, pero cuando hacíais el screening de RNA interferencia, ¿cuál es el fenotipo? Se dice, seleccionamos los genes que daban más de un 10% de fenotipo. ¿Qué fenotipo?
Sí, me alegro que me hagas esta pregunta porque a veces, como estás muy acostumbrada a hablar de lo mismo, parece que es muy obvio y no. Nosotros lo que contamos es el número de neuronas, porque como veis, el gusano, una vez que se ha acabado de madurar, todas sus neuronas mantienen la misma posición. Y como veis, esta población de neuronas son en total unas 16, son bilaterales, pero puedes perfectamente contarlas porque además primero, el reporteo que nosotros que utilizamos es fluorescente y es que además el gusano es transparente. O sea, tú puedes ponerte como unos 30 gusanos en el microscopio y contar cada uno de ellos si llega a sus 30 neuronas monoaminérgicas o no. Y eso es lo que nosotros utilizamos para tener el fenotipo, si hay fluorescencia o no.
Si hay fluorescencia de...
En esas neuronas.
¿Pero de toda o de algún subtipo en concreto?
De todas. Puedes medir niveles de intensidad, pero para hacer el screen nosotros no queríamos ser tan precisos y contábamos si de cada una de ellas, por ejemplo, ADF, que es la última de la que os he hablado, hay 2. Si hay 2 ADFs o no hay. Y representamos, digamos, el total de una población que contamos que son 60 gusanos y de esos 60, si tendría que haber 120 ADFs, ¿hay las mismas o hay menos?
¿Alguna pregunta más?
Yo te voy a hacer una pregunta. Primero, ¿por qué os habéis centrado en ADF? Supongo que habréis empezado por ahí, tendréis pensado mirar las otras neuronas. Y segundo, me ha interesado mucho lo del calcio, porque a mí me gustaría hacerlo y cuéntame un poco cómo lo hacéis, cómo medís.
Pues nos centramos en ADF porque fue de los primeros candidatos y aparte porque es otro de los proyectos que está llevando a cabo en el laboratorio Miren Maicas, que es una postdoc de nuestro lab. Y luego el calcium imaging lo hice en una estancia en la que fui. Lo que hacíamos era utilizar un sistema de microfluidics y al final tenías una microcámara donde ponías a los gusanos. Entonces, controlando el flujo, eras capaz de hacer pasar una solución basal. Los gusanos estaban inmovilizados, pero cada vez que le íbamos a dar la estimulación hacíamos pasar la solución de estímulo y cuantificábamos la fluorescencia de la activación que provocan estos químicos, porque la neurona expresaba una proteína que es sensible a calcio. Entonces, cuando se activa la neurona, hay un aumento de la fluorescencia. Básicamente funciona así. Y cuantificábamos si aumentaba más o menos comparando el control con el RNAi.
Entonces, claro, tienes que tener alguna cepa con algún marcaje fluorescente para hacerlo.
Sí, hay proteínas o fluoróforos que se utilizan ya establecidos para hacer el calcium imaging.
Muchas gracias, Ángela.
¿Y la técnica que has comentado del calcio se puede utilizar aparte de gusanos? ¿Sabes si se utiliza en otro tipo de...?
En todo. Realmente es parte de la optogenética. Todas estas técnicas de control a través de la luz de proteínas que se vuelven fluorescentes, entonces se utilizan células, en ratones, en todo. Está bastante establecido.
¿Más preguntas? ¿No? Pues siguiente...